The COVID pandemic has brought more media attention to clinical laboratories than at any time in recent history. Today, we'll talk about media coverage of diagnostic testing for COVID-19 with two experts. Some of the questions we'll discuss include, how has the media coverage of tests for COVID-19 affected the public's view of these tests? What is the role of social media in talking about testing for COVID-19? And how do reporters covering COVID-19 testing work with clinical microbiologists to get the story right? Welcome to Editors in Conversation. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org and on Twitter @jclinmicro. I'm your host, JCM Editor-in-Chief Alex McAdam. This podcast is supported by the American Society for Microbiology, which publishes JCM. I'm joined by two expert guests. Dr. Katherine Wu is a reporter for the New York Times covering health and science. Dr. Wu writes about a broad range of subjects in science and medicine, and she's published many articles about tests for COVID-19. Thanks for joining us, Katie.
Great to be here.
And we're also joined by Dr. Susan Butler-Wu, no relation, right?
That we know of.
We know of. Dr. Butler-Wu is an associate professor of clinical pathology at the University of Southern California and director of medical microbiology at Los Angeles County and Medical Center. Dr. Butler-Wu is also a member of the editorial board of JCM. Welcome, Susan, and thank you for being here.
Thank you for having me.
Let's start by talking about some controversies around testing that have played out in the media. First, there's been a lot of concern about reverse transcription PCR and the other nucleic acid amplified tests. I've seen articles saying that they're too sensitive, that false positives are common, and criticizing the test because it does not detect live or replication competent virus. I'd be curious to hear from each of you about why there's so much concern about RT-PCR. Susan, why don't we start with you?
Yeah, so obviously, pre-COVID, we've been dealing with a lot of conspiracy theory things over the years with respect to various infectious diseases, and so COVID has not been immune from that. So there's this sort of narrative out there that somehow there's a false positive issue with PCR. And so, I've seen things saying like it's because we're running 45 cycles, and the more cycles you run, you're going to pick up stuff that's really not there. And as a scientist, that's very frustrating because we don't pick up stuff that's not there. That's just not a thing. And so, with respect to the idea of false positives, as somebody who works in a clinical microbiology lab, when we detect SARS-CoV-2 RNA, you may not be symptomatic right now, but you have had it. Either you had it a couple of weeks ago or you have it now and you're asymptomatic. So that's been one of the very frustrating things as a clinical microbiology lab director. And so this idea of this case-demic, it's a myth. We don't amplify stuff that's not there unless there's a false positive that occurs by introducing RNA through a technical error. That's extremely rare. And the second thing is we know that the numbers match what we see in the hospital. So when we were surging, we had lots of positives. We came way down. We had very few patients in the hospital hospitalized. And now, yet again, we're starting to surge. We see those numbers of detections go up and the concomitant increase in terms of hospitalizations, people going to the ICU. So they track. And I think we just, as scientists, we just have to keep bringing that to the fore, that this is not a case-demic. It is not a false positive. If you test positive, the vast majority of times, it's really because you had or have COVID. And in terms of the persistence of RNA, we've never, as diagnosticians, had tests for active replicating virus outside of culture. And nobody wants to go back to culture again as a diagnostic method for virology. So.
Thank you very much, Susan. Katie, what would you add about how RT-PCR has been discussed in the news?
Yeah, I agree with everything that Susan just laid out. I think it's so important to highlight how reputable PCR is and how long it's been used. It's not this brand new thing that people just brought in for the COVID pandemic. It's been used for so many things. It has a fantastic track record. There's a reason that PCR is a fixture of just about every molecular biology lab out there, regardless of whether or not it has anything to do with infectious disease. It's just this excellent technique. But in covering tests that use PCR and seeing other people cover tests that use PCR, there has been a lot of vitriol tossed around about it. And I do think, the crucial context here is that people are incredibly frustrated. They're scared. They don't know what's going on. They want to know if they have this virus or not. And it scares them to give a sample that might have been really painful for them. If that swab goes all the way back into their throat, then that swab disappears, and some occasionally indeterminate amount of time later, they get a result back and they don't know what happens in between. I think when people are so frustrated, it's very easy to criticize, but exactly as Susan said, it's so important to make sure that the science of the story is getting out there and make sure that people aren't just thinking, oh, it's too slow, it's not good enough. But there are people working incredibly hard to make sure that these tests are giving back accurate results, and we should rely on them.
I want to push on this a little bit, and this is a tough question, but where do you think, either of you, where do you think it comes from, specifically the concern that that PCR does not detect replication-competent virus? How has that come into the public consciousness and the media, and social media? Why is this such a thing? Susan, you want to take a stab at that?
Media, Katie, if you want, maybe you go first.
Sure. I think this actually does come down to a couple things, and one thing is timing. We all know that if PCR is running very smoothly, it can turn around and answer in a few hours, and that's amazing. That is still happening in some cases, especially in places that have means and resources. But the truth of the matter is, this pandemic is at the point where the virus is so out of control that demand for testing has exceeded capacity for testing. And that is not the fault of the hardworking people in labs, nor the fault of the lab's infrastructure necessarily, but really just the fact that we're in this unprecedented situation. But knowing that there are people out there who are waiting days to get their results back, I think there is, something to be said, and it's a nuanced thing, that it is really hard to interpret a result that comes back 4 days later, but is reflecting the state of your health like 4 days ago. People don't know what to do with that result. They're also being told, disregard that result. It may no longer be true. And that's incredibly confusing. They waited a long time for this information and they're now being told it's useless. And so I think that prompts a reaction from people. Certainly media reports haven't helped this, saying that, yeah, the longer you wait for that test result, the less useful it becomes. And I think there is room for a scapegoat to enter the picture there. That's one huge part of it. And we also know that, if you even look at CDC guidance, it does say, part of the reason that you may not want to retest within 3 months of a positive result is, that one, reinfection is pretty unlikely, very, very unlikely in that amount of time. But also we do know that in some cases, nucleic acid could linger, and it's not always clear what that means in all cases, because again, we can't test for infectiousness in a straightforward way.
And we've never tested for infectiousness really in the context of modern diagnostics. And so I also think that we have to understand that from the get-go, the response in the US has failed. It's failed from the top to the bottom. And so, I also think with respect to expectations of people, right? People rightly have this expectation that they should be able to access testing, that it should be back quickly. It should be the best test that you can get. And so, but we really have this patchwork quilt of basically everyone fighting for themselves to be able to set up testing in their facility. So I think it's just this nexus of, fear and a lack of appropriate response at the government level. And it's all mashed together to create this. And this concept of that's why I think this idea of being false positive and, I'm detecting it, but I'm not really infectious. But you were infectious, right? You might not be infectious now, but you were infectious a week ago or 2 weeks ago. So I don't really understand why, this idea I don't want to know, it's not useful to me. Well, you had COVID. I mean, that's an important thing to know. And so it's interesting.
I think one other thing to maybe tack on here, and this may actually be a good transition into our next topic, is that PCR is obviously not the only option available for processing a test at this point, because people have come forward with alternatives that have so frequently been billed as, what about a test that's faster and may even be better suited for the needs of this pandemic or your personal needs, a quote unquote rapid test, an antigen test, a test that circumvents the laboratory. I think when people see solutions like that, it's very easy to create a false dichotomy of like, here's a new thing that will fix my problem, which means the old option, PCR, was not a good test. And that's not correct, but that's been hard to communicate because people are used to progress and catching on to flashy things that promise solutions to problems.
So is it sort of that people see antigen tests like the hero on the white horse riding in to rescue us from the dragon of PCR?
I think sometimes, yeah, because it has been kind of framed like that. Like I can't count the number of articles that I've seen that have started with PCR, it's slow, it sucks, like it takes forever to come back from the lab. But wait, there's a solution that will fix all your problems. Like it's fast, it's awesome. It will give you your results right away. And it'll also tell you this and that. I think people like reading those things, and I totally understand why they're written that way. It creates stakes, it makes a piece more readable, but it also does contribute to this idea that it's like PCR versus not PCR, when really what we need is, a more catch-all approach where test techniques are complementing each other, not fighting each other.
Exactly. And there's, the right, what's our mantra in lab medicine is the right test in the right patient at the right time. And so all of these are ways to basically diversify our ability to reach patients, but they're being pitched as like you said, Katie, that the PCR is terrible and antigen is going to save the day, which I find really interesting because I think about, how prone to litigation we are in this country. And physicians are practicing medicine from a place of, defensive medicine. But at the same time, there's these calls for these tests that we know will miss some people who, even if there's no question they're infectious, it'll miss some. And yet there's this, groundswell of like, we have to have these. And so just the more conversation we can have, in a balanced way where people fully understand the pros and cons of each approach and that are educated about the test that they're getting, and that there's no test that's 100% one way or the other, and that they understand that. I think that the more that conversation can be happening in the media and elsewhere, the better. There is no magic solution to this. We are not going to just test our way out of anything. All of these things are like, Dr. McKay's model. Testing is one piece of the Swiss cheese. And I think that we have to remember that and educate people about the pros and cons of the different tests that they're getting.
So, Katie, how do we go about that? How can the media best portray the diversity of tests, the advantages of the various tests? Because it seems like, exactly as you said, the story is usually, here is the test that's going to solve the problem of testing. You can do this one at home. Well, then it turns out that it's not sensitive, or it's not specific, or it's not affordable, or you have to have a prescription for it, or something like that. But how can the media sort of present a balanced point of view about the various tests?
I don't think I have a perfect answer to that. I'm not sure one exists. It's incredibly difficult. I think in a crisis, people don't feel like they have time for nuance. I mean, they don't. People have their lives at stake. They have their livelihoods at stake. They want a solution. They're craving a solution. And I think people are trying to wrap their minds around, there's not one silver bullet. Like, it's not only testing. It's not only masks. It's not only physical distancing. But it's appealing to hear something or read something that promises, like, here is a huge, game changer is a word that's used every day in this pandemic. Yeah. People want to latch onto that. It really helps them. I think, unfortunately, the truth is less flashy than that, but I hope that people in the media are coming around to this idea that, we should not be pitting tests against each other. I don't ever want to portray myself as team PCR or team antigen. That is not a dichotomy that should exist. I think both types of tests are fantastic. They both have enormous benefits, and they can both solve specific types of problems. I think the problems come when people portray them as opposing forces. And so I really hope to see more balance in the future and more acknowledgement that one type of test is not going to be, the cream of the crop that rises above the fray, and testing itself is not going to be the fix-all that people need. I think it just needs to be like a sort of balanced breakfast portrayal instead of here's your quick fix.
Yeah.
So make sure to have so go ahead, Susan. Yeah.
Oh yeah, I was just going to say, I think too, one of the nuances that have been missing in a lot of the narrative around testing is that it's not a single solution. And I think we need more conversation around the fact, like you just said, part of a complete breakfast, but it's not the complete breakfast. And too often, I think, people are, there's sort of this sort of narrative that this kind of passport, like I'm clear to go do something because I'm negative, without fully understanding, like that's part of it. And I think that conversations around testing and how we can use them to get a handle on things and to turn the course have to be very, very clearly, emphatically accompanied with very strong statements about how they have to be accompanied with all the other things that we work, or they're not going to work. I view it like in the clinical lab, we do rapid diagnostics for blood cultures. If we just do them, but we don't combine it with anything, it accomplishes nothing. If the person doesn't act on it appropriately, if it's not communicated appropriately, it fails, even the fanciest test in the world. So it all has to be combined to have that impact. And we need more framing around that in the media, really getting that out there.
Let's talk about how serology testing fits into this complete breakfast. It seems to me, and I'd be interested to hear what your thoughts are, but it seems like early in the pandemic, there was a lot of confusion, even in the clinical literature, about the place of serology for a diagnosis of acute COVID-19. Most of that seems to have been cleared up, although I'd be interested to hear what you think about that. And then more recently, there's been some discussion about the FDA's emergency use authorization of a test specifically for neutralizing antibody. And I thought the media coverage of that was interesting as well. Susan, why don't we start with you? How do you see the media view of serology, and anything specific about that neutralizing antibody test?
Yeah, so in terms of serology, I mean, definitely it was that the conversation was initially framed as, the whole concept of like, can we have a test? I guess is at the core of the same thing. Can we have a test that allows one to assume it's safe for you to go about your business without any of the other measures, right? Fundamentally, like, am I immune? And so, the WHO came out and said there's, really immunity passports would be a terrible thing. And so there was this great excitement about serology that then in time waned. And now pretty much, at least where I work, it's kind of, we use it for kids with the Kawasaki-like syndrome. We use it for folks where, they're a couple of weeks into their illness and maybe their PCR is negative. But in terms of it just being this widely thing where everyone knows their status and then carries on as if they're immune, we know that that's not what's recommended. And even if you do have antibodies, that you still need to protect yourself and protect others. In terms of the neutralizing antibody, there has been, like you said, some interesting discussion in the media. Again, I'm kind of sitting back with the popcorn and just like waiting and seeing what happens with that. Kind of like happened with serology. I mean, we're at a different point now. Obviously, the pandemic is raging. It's just raging. It's a full, out of control wildfire in this country right now. And so I'm like, okay, cool, neutralizing antibodies. Let's see. We're looking now, knowing that vaccines are coming, which again, we're going to have to be combined with other measures. So honestly, Alex, I'm kind of like just sitting back and waiting to see how it plays out. I think, latch onto it as the great new thing. How's it going to change things? But it's not clear to me how it is really.
I'm going to opine about this, and I want to hear, Katie, what you think about this. To me, the coverage of the neutralizing antibody neutralizing antibody test that just got emergency use authorization. The coverage seemed a little breathless in the sense that there were lots and lots of articles, which all looked very similar to each other, saying that this new test was out there, pointing out that it would detect neutralizing antibody, that is antibody that prevents the virus from being able to infect a cell, and distinguishing that from binding antibody, which all of a sudden looked second rate, that detecting binding antibody was not really up to snuff. And then the articles, for the most part, stopped there. But what they did not explain, and what I'm not sure I know, is why that is useful or significant. It might be useful for a vaccine trial, for example. I don't know. But I have the impression, you're going to have to correct me, Katie, but I have the impression that the company that came out with the test did a great job of capturing the media around that test. And there was article after article, as I said, describing the test, but not really talking about potential significance of the test, which is still, I think that there will be significance to the test, but I don't know what it is yet. So So Katie, was the media hoodwinked around this test?
I mean, I will say, like, when those EUAs came out, it appeared in our New York Times Slack, and we decided to pass on it. But some other outlets definitely did cover it. And I think I agree with you. There was not enough context, but it was definitely portrayed in almost a press releasey fashion. Like, look at this, again, game-changing new test that is going to, give you hints about immunity and your degree of protection against this virus. But there was a lot of context missing from that. I mean, I think what's crucial for people to understand is that neutralizing antibodies are a correlate of protection, and we all know correlation is not causation. There's confusion around that. And I think part of the problem is neutralizing antibodies have been championed in the media for months now as like a crucial factor. These are the antibodies that can block the virus from entering a cell. They can stop an infection in its tracks. Maybe your cells won't be infected at all. And I think after so much conversation about vaccines, and now that the tone of the pandemic has really shifted to be around vaccines, I think people were excited to see a test that might give them a sense of like, well, am I basically already vaccinated or immune, even though these tests can't really tell you that. I'm glad you sort of framed antibodies the way you did, because I do want to give a shout out to binding antibodies, which are awesome. They're not just like filler in your burrito. Like, they're there for a reason. They do lots of great things. Our immune system would not be the same without them. Like, we need antibodies to sequester viruses, to coat infected cells, to activate complement, all those wonderful things. It's not just about neutralizing antibodies, and we don't fully know the full contribution of neutralizing antibodies. And I personally don't see a ton of general utility for these tests yet. I'm happy to be corrected at some point, but I really hope that people are not going out and taking these tests and declaring themselves immune. That should never be the outcome of any test, no matter what it's measuring.
I think that's what Susan. Ultimately, right? Like they want, it's that concept of, I am like now in a position where I'm safe. I mean, and that's the danger here, right?
I want that.
We all want it.
We all want that.
And what are real or different.
Yeah. Anything else about antibody before we go on? All right. If not, the other media in which COVID-19 is talked about a lot is social media. All three of us are on Twitter, which is my way of saying, as a 55-year-old, could we please stick to Twitter, because I don't understand the other ones. I don't want to talk about TikTok. I don't know what it is. But Susan, let's start with you. You're very active on social media. All three of us are. Susan, how would you characterize the discussion about COVID-19 testing on Twitter? And you can talk about other social media if you want.
A dumpster fire. It feels like a dumpster fire at times. It's a fantastic medium to learn. And I definitely see papers that I just would probably miss or I may discover in 6 months. So there's real pros and cons. But, definitely there's that thread of like a lack of stability, a lack of cordiality, a tone of discussing things with people that is different than what we're used to in science, where, we are professionally talking to colleagues, and we understand that as scientists, we may have opposing viewpoints, and that's okay. And we debate the pros and the cons, and that's our job as scientists. And so, Twitter's hard because I feel like there's a definite element of where that kind of goes out the window. And I don't know if it's I can't blame it on the character limit, because there's threads. So I think it's a real, like, there's really two pieces. I love how much I learn from it. And then there's times where it's toxic and I have to not be looking at it because it stresses me out. And so, some days it's more one or the other, but it is a fantastic way to learn. It's a fantastic way to connect with colleagues and to connect with people like that you otherwise would never really come across. I guess. I just lament the inability to just, cordially disagree with each other, and that be okay. And then, like in the conference, you disagree and then you go for a beer. It's like there's just a disagreement and there's no beer.
Yeah, I think universal beer would solve a lot of Twitter disputes. That's deeply unfortunate. Yeah, I agree with all of that. And like Twitter, all social media is such a double-edged sword. And honestly, recently, since the start of the pandemic, the stressful part of that double-edged sword has felt a lot sharper. People are very, very, very stressed. And I think there's a couple things that come to mind when I think about social media. One is like, there's a degree of anonymity when I just type something into my computer and I send it out into the universe. There is so much distance, both physically and virtually, between me and the people who follow me, the people who see my tweets. If I say something, I can delete it later. If somebody is mad at me, they're not going to come to me and yell at me. That their anger is represented as words on a screen. People are often bolder on Twitter. People love getting angry, and it's a place for people to vent. And I think that does fuel a lot of conflict. I wish people were more civil and allowed for more nuance, but it's just not the right venue for it. It's too restrictive. It's too anonymous for that to happen. I think one big problem with that is so much of the scientific process gets lost when science is communicated on Twitter. Like so much of science is we don't know, or, to be determined, or we need more data, or like this, but also this. And there's just not enough room for all of that. That's seen as fluff, or like get to the point already. Twitter is unfortunately the place for punchlines without caveats. And I think that has created a lot of confusion in some circumstances. And, people type on their phones and they post without thinking. It's just, when you don't have the time or the space to be careful and nuanced, people get angry very quickly and things just escalate. So I try to stay off Twitter late at night when things get a little crazy, but otherwise, it's a necessary evil. And I will reiterate what Susan said. It's such a nice place to find people. Like, I found sources on Twitter, including wonderful clinical microbiologists that I see tweeting, and I'll just message them and say, hey, I really appreciate what you said. Do you want to talk sometime? And that's been wonderful.
So it's a great way to find people. What else is good about Twitter? We're all on it, right? You guys probably checked it twice during this conversation.
I have not checked.
I haven't either. I'm just kidding. But why do we do it? What's good about it? What are we getting out of it?
It is a really quick way to get information, not just, to meet people, but I mean, people will post on Twitter like, hey, new preprint, check this out. Or like, I made this graphic, or, we just cured this mouse of type 1 diabetes in the lab. Like, awesome. And for me as a journalist who has to keep up with breaking news and the latest developments and wants to find new angles on things, Twitter can be a gold mine, but it's unfortunately a gold mine filled with a ton of coal and rubble. And sometimes it takes forever to sift through the extra stuff to find the valuable information. But that is where it often appears first. And so I think I do have to invest in it. Twitter can also be wonderful. Like, I have seen threads of people like being incredibly supportive of each other and shouting out other people and saying like, I'm so grateful for Susan, or like someone else who is working incredibly hard or putting the right information out there. Like, people can be supportive, and social media, I think, has been a really wonderful outlet for people during a time when we have to be physically distant from each other, but Twitter's a way to stay socially close in some context.
Definitely. And without, we don't have conferences now, or even virtual conferences, the same, right? You're not like walking through and you're like, oh, hey, Alex, how's it going? You don't have that. So to me, I agree, Katie, that it's a great way to stay connected with people. And if I think back to, my brain can go back as far as pre-COVID, definitely in terms of research and papers coming out, that's where you hear it first. And there's definitely studies that I've come across through Twitter that I may not have on my own. And I think that's a really, really major advantage. I do think, though, just back to the nuance piece, and sort of how it all connects. When we think about what we do in clinical microbiology, there's so much gray to what we do, right? Like, this organism from this patient may be infection, it may not, right? We're constantly interpreting stuff. And so that is the problem with Twitter, and just society wants black and white answers around this. And there aren't a whole lot of black and white answers in medicine, and certainly not in lab medicine. Really in lab medicine, there's not. So that lack of nuance is a problem. But overall, I mean, if it was that terrible, I wouldn't be on it. So there's more good than there's bad.
You can always turn it off. I'm surprised how often I see people saying, people who are very active on Twitter saying, bye everybody, I'm shutting down for the weekend, or I'm shutting down for the rest of the month. There is this feeling that we need to unplug from it or detox from it. I certainly take weekends off from it sometimes because it gets to be too much.
Totally.
The constant checking. Yeah.
Yep.
Susan, I want to change subjects a little bit. And this is a slow pitch question. How do you think the media coverage? I don't know
baseball metaphors, by the way, because I'm from Ireland, so I don't know what that means.
Well, we'll see what you make of it then.
Okay.
How do you think that the media coverage of the COVID-19 pandemic has affected people's awareness or their view of clinical microbiologists and our field of clinical microbiology?
So again, not understanding a slow pitch question, but I think in terms of the question, I think it's been a really positive thing in terms of bringing attention to the fact that there's people like us and that there's clinical labs and there are people who are experts in lab testing. I mean, often as clinical microbiologists, everyone wants to talk first, right? We've got various physicians, pharmacists, everyone wants to talk for what we do, but nobody knows what we do like those of us that do it. And so I think it's been really fantastic that there's been clinical microbiologists out there in the media talking about what we do, bringing attention to it. I had an opportunity to be in a class with Dr. Juliana Lemieux, her class that she teaches in New York. And then definitely there's a bigger awareness of testing and that, this could be a career amongst people. So I think that that's a really positive, one of the few positive things that's come out of all of this is understanding that this is a career and it's a rewarding career and that there's people with expertise in this and this is the time to listen to them because they know what they're talking about. So that's been a really positive thing.
I think so too. And I've found that I much less often have to explain what I do to people. Right? It used to be that I would say, I'm a clinical microbiologist. I do the diagnostic testing in the laboratory at the hospital. But now people actually know that we exist, right? People who I've known for years who sort of knew I did something at the hospital, something in a lab, all of a sudden they'd say, oh, you're that person, you do that.
Yeah, no, that's been a really positive thing. There is less explaining what we do. And it's funny, after years of my mother still not really understanding what I did, I think she gets it now. So, yeah, that's a good thing.
So Katie, let's turn to a little bit about what it is that you do. And this is a very broad question, but how do you approach your articles about COVID-19 testing? How do you initially find the subjects to write about? How do you decide whether they're important or not? How do you find the experts who you talk to? You've touched on a couple of these things already, but can you kind of bring it together for us? What's the process around an article?
Yeah, I think one thing that's been really interesting is over the course of this pandemic, my approach to writing about testing has changed a lot. In the beginning, people didn't even really know what tests we're doing or what tests were. And I think one of the first pieces I wrote on testing, I interviewed you actually, Alex, just to explain like what happens when these tests are processed in the lab, what they're actually looking for. It was a very explanatory, here's what you need to know about testing sort of piece. And in the months since, I think just the nature of the beast, testing has become a little more polarizing. It's become a little more controversial. People have come up with unusual ideas about testing, some more true than others. And a lot of what I do now is try and figure out what are the misconceptions around testing, or what are the questions that people are asking? Are people misusing tests? Are people misunderstanding tests? Can I shine a spotlight on that and bring some more clarity to the picture? Can I drill down on what the most newsworthy tests are? And that can either be a new test that's gotten an emergency use authorization, or it can be a test that's being painted in a new light by a researcher, or it has been validated and yielded surprising results. I think especially during this pandemic, I'm always trying to find stories that will contain information that will hopefully affect behavioral change in a positive direction. So if people better understand their tests or know how to understand the results, then I think I'm doing my job right. But another crucial part of my job is figuring out, is there corruption? Are people abusing their power around tests? And that's not ever a realm I thought would ever intersect, like diagnostic testing and like crazy abuse of power. It's not like we're dealing with some sort of Watergate scandal in testing right now, but there are things out there that have gotten controversial. And I think that's fascinating as well. Like, people have agendas around testing. And I think those are important to unpack and clarify for people. Here's why some people might think this, but remember that there is scientific backing. Here's a fact. Here's an opinion. Don't conflate the two. I was just going to add, like, in terms of finding experts for that, because that was part of your question. That's tough. A lot of people have waded into testing in recent months, which I think is so interesting, because it's not easy. It's so complicated, and there's so many concepts to understand. I certainly don't know anything, which is why I need to call experts. There are people who are very used to doing diagnostic testing in the clinical setting. Testing is now also taking on a really crucial public health role. So I try and talk to people from both sides, but I find experts on Twitter. I find experts by talking to experts I already know and asking, hey, who else should I talk to? And also just seeing who's being really vocal about testing or who's asking questions about testing. Other crucial sources for testing include like people who don't have expertise in testing but are getting tested. Like, I want to hear from those people too.
Let's talk a little bit about sort of a narrow subject, but one that, frankly, as a journal editor, I'm very conscious of in this area, and that's preprints. Preprints, as you both know, of course, are articles that have been posted by authors on publicly available websites like bioRxiv or medRxiv, and they're posted before the paper has been accepted for publication in a peer-reviewed journal. So, Katie, how do you fit preprints into the media process? How do you distinguish between those and peer-reviewed articles?
This is a great question, and it's caused like a little bit of controversy because, the whole like peer review is so critical. The reason we have peer review is so that, the majority of published articles that are appearing in scientific journals have been vetted. They have the right controls. They're framing the results correctly. You don't get that with a preprint. It could literally be someone typing at a computer with poorly controlled data and just putting it online. And there are no ratings on preprints. You can't distinguish those without a trained eye. So I do cover preprints because I think, especially during this pandemic, it's become so necessary to get the information out there quickly. Publication takes so long. It can take years sometimes, and people want to know about the virus right now. But every time I cover a preprint, I am really careful. I will contact like extra experts to make sure that they're vetting it. And, sometimes we will literally sit down with the manuscript in front of us, virtually, of course, and like literally go figure by figure and peer review the paper ourselves to make sure that I'm not covering something that's amiss. And I'm always perfectly happy to cover a preprint with questionable findings, especially if it's like going viral or someone has misinterpreted it. Sometimes I need to debunk a preprint, and that's also really valid and worth doing. I think one thing that's critical when covering preprints is always making sure the reader knows it is a preprint. And hopefully more and more readers over the course of this pandemic have started to understand the difference between a paper that has been posted to a preprint server and a paper that has been published in a peer-reviewed scientific journal. Definitely not everyone knows that, but I hope that number is more now.
I think you've done it. I mean, in terms of like, oh my gosh, but you've just done a fantastic job. Really, we're so lucky to have you doing what you're doing, because I think you've been really just effective at doing exactly that, at covering what's out there, what's controversial, and making it very, very clear, like what the caveats are. And so just kudos to you for your reporting on this and for tackling preprints, because pre-COVID, I think we listen maybe a little bit more to preprints now, and certainly they're out there in the global sphere. So it's really critical that you're doing what you're doing and going through that process to get the information out there. So thank you.
Oh, thank you.
Agree. I have to say, as a journal editor, there is a special type of pain that only journal editors know when a preprint is out, it is covered in a very prominent media source, Washington Post, New York Times, whatever it may be, and that paper is under review and about to be accepted and published, but that's not represented in the media. Oh, it hurts. I can't tell you how much that hurts. All right. Let's see. Susan, last question for you. I think it's fair to say that you have a great way with words, and we've seen that during this discussion. And that, along with your great expertise in clinical microbiology, has made you a valuable spokesperson about COVID-19 testing. Tell us a little bit about how you interact with members of the media. I see you frequently on the news, in articles. How do you decide who you'll talk to? Do you do any preparation for the interviews?
This is just very surreal to even be ever in a position to ask this question, to be honest. And I will say I'm not the sole person. There's many of us have been in the media. So how do I interact? I get emails. I loop in our media person and I talk, and everybody I've talked to has been really, really great in terms of preparation and my notable comments. I don't really I don't pre-think what I'm gonna say. I don't I would liken this to maybe it's being from Ireland and allegedly born with the gift of the gab. I don't think about analogies. I mean, Katie I just talk and then some words come out, and some of them are probably crap and some of them are useful. In terms of preparation, certainly if it's about a paper or, a topic, I would do some reading about that just to make sure because things are moving so fast in COVID land. I mean, what I know now, I may have already missed like 4 papers from yesterday. So I would definitely do that sort of thing. In terms of who to talk to or who not to talk to, I pretty much talk to everybody because I think it's an opportunity to bring attention to what we do as clinical microbiologists. And certainly I share the names of other clinical microbiologists with journalists that I talk to, because often journalists will say, who else should I talk to? And so there's, I have a list of other wonderful folks in the field who have different perspectives, and, no one clinical lab is alike, and no one clinical microbiologist perspective is the same. So I don't do a whole lot of prep other than reading whatever papers I may have missed that are germane to what we're going to be talking about. And that's about it. I pretty much just, and that's it. Yeah, for better or worse.
Anything else we should talk about? Anything we've missed?
Oh gosh. How are you both feeling at this point in the pandemic about media coverage and the visibility of your field?
Alex, do you want to go first?
Sure. I feel, we were talking about this a little bit before. I feel absolutely great about the way clinical microbiology is all of a sudden something that people know exists. And I hope more broadly it'll affect our friends in clinical chemistry and hematology and so on, because we have been, doctors helping doctors, but people didn't know that we were out there, and they didn't understand the complexity of what we were doing or how interesting and fun what we do can be. So I think that piece of it is absolutely great. The media does a very good job covering clinical microbiology and COVID-19 testing, I think, for the most part. Certainly there's the occasional article that makes me want to tear my hair out, but there's just a tremendous volume being written right now, and I think that's perfectly understandable. So I feel, the COVID-19 pandemic is an absolute disaster. It is a horror. But just this little piece of this little glimmer of light, I think, has been a good thing for clinical microbiology. Susan, what do you think?
Yeah, I agree. On a personal level, it's really rewarding to just be, listening to NPR on the way home and I hear Melissa Miller, or, you're exactly. Like, oh, I know this person. And this is so great that they're bringing attention to what we do. It is probably the only ray of a glimmer of light, as you said, Alex, in all of this. I think, that's been definitely the one positive out of all of it. In terms of other things, though, sorry, separately, you said anything else we need to talk about? I think that the home testing piece, we need to sort of continue to bring attention to what the limitations are with that, so that the public understands, what it means or doesn't mean in that context. And that even though testing is going to move into the home potentially, as clinical microbiologists, we have the expertise in diagnostic testing. And so we really need to be educating people about what things mean and don't mean as we move forward.
Yeah, I certainly agree. Well, thank you, Katie. Thank you, Susan. This has really been a lot of fun. I appreciate your time very much.
Thank you.
Thank you for having us.
You can find the Journal of Clinical Microbiology at jcm.asm.org. Thanks for listening.